Yes. Okay, so good out. Okay, just a minute. Yeah. So good evening. Welcome to today's session. This will be our 22nd class. We have 3 more classes left. The next class will be tomorrow, and the next week we will wrap up all our classes for this course. So, if you remember, in our previous discussion, we had started our discussion on high throughput sequencing techniques to study the conformation of DNA to understand that chromatin structure, chromatin accessibility, and the way in which DNA is organized makes a lot of difference on whether or not it can be expressed, to what extent it can be expressed, and um, the kind of control that can be executed on the DNA. So, in the previous class, we had discussed attack sequencing, the introduction to attack sequencing technique. In today's class, we will look at 3C methods, 3C, 4C, 5C, some introduction to QIAPET, etc. And in the next class, we will, which is tomorrow, I'll continue it a little bit forward, and then we'll move on to our discussion on genomic and epigenomic markers, biomarkers in clinical diagnostics and prognostics, with an overall summary of everything that has been covered till now. And in the next 2 classes, in the first class, we will have an exam because just to wrap up the entire discussion till now, we'll have a test. And after the test, we will summarize multi-omic studies and the concept of multi-omics. And the last class will be for discussion, which will summarize the entire course. So that is the whole idea. So with this in mind, we will first quickly recap what we have already discussed in the previous class about chromatin accessibility. So what does it mean? It means how much of the chromatin is open or available for recruitment of transcription factors, chromatin remodelers to open or close DNA. To understand that a complex formed by DNA and associated proteins is not just a linear structure, but rather a three-dimensional structure, which includes histones, transcription factors, chromatin modifying enzymes, and chromatin remodeling complexes. So traditionally, it is probed by assays that involve nucleases, mostly that is DNase, which is an enzyme that digests DNA, or using restriction enzymes, typically are able to work at few selected genomic regions every time. And one of the first methods or experimental strategies that was used to understand these chromatin accessibility understand chromatin accessibility was published using DNase 1 digestion, and the technique is referred to as DNase-seq. So in DNase-seq, you isolate nuclei, you permeabilize, you do that by permeabilizing, you follow that up by permeabilizing the nucleus using detergents like Triton X100, such that DNase enzyme has access to the DNA which is inside. After digestion, the fragments can be then purified and size selected for library construction and subsequent sequencing. Because this process is enzyme dependent, how much of the enzyme is available can make a lot of difference, and therefore the data quality can also get affected accordingly. Therefore, the amount of DNase 1 sometimes needs to be titrated to achieve optimal activity when using a new type of cells, or when DNase 1 is used from different manufacturers or different companies. DNase-seq has also been carried out for FFPE samples, which are formalin fixed paraffin embedded samples. And this is an example, a schematic sort of, which represents how this process exactly is done. So if you look here, this is your heterochromatinized or sort of histone-bound form of DNA, which has certain regions which are accessible because transcription factors have been recruited there, chromatin has been remodeled there, and that region is of interest, and therefore its expression is being something, its expression is something crucial for cell survival, and it is being regulated by the cell at multiple levels. And following that, you can see that these are the reads which represent that these regions are accessible, and they justify why that region is is getting coded. So following, oh, sorry. So following that, we understood that attack seek, which is an sort of an upgrade or an advancement on this previous technique, is a sort of an alternative strategy which allows you to do something similar. It allows you to understand how the profile of chromatin accessibility in a cell looks like, or what it looks like at a certain point of time. So one of the major differences between this technique and others is that the T in ATAC-seq stands for transposase. So this transposase is, of course, an enzyme which, if you have studied about transposons before, you might be familiar with. Is the enzyme which is crucial for that process. And the transposase used for ATAC-seq is genetically engineered. And it is a hyperactive transposase, TN5 transposase, that is preloaded with adapters that simultaneously cleave, and it has tags which are accessible, or nucleosome-cleaved chromatin regions are present. So if you look here in ATAC-seq, you have this TN5 enzyme, which has these two sort of red and blue colored different adapters, which we'll learn about. So this TN5 enzyme is able to cut and is able to sort of tag or mark the region that it has cut. And that is how the annotation is sort of helped out. So what happens in this process? You once again, isolate nuclei from cells, the chromatin structure, and any other associated DNA binding proteins, including nucleosomes and transcription factors, are kept intact. Then the chromatin is subject to treatment with TN5 transposase, which acts like a homodimer that allows it to simultaneously fragment the chromatin and insert certain sequences which enable downstream amplification, which are these two types of adapters. One is the I5P51, the other one is I7P7. These are these are ATAC-seq adapters. Only those fragments that receive one I5 adapter at one end and the I7 adapter at the other end will get amplified and undergo sequencing. And after analysis, genomic regions or genomic fragments which are enriched for many transposition events will be recognized as ATAC-seq peaks, and that will give you your conclusive, statistically significant, and biologically relevant conclusions in your study. So this is what the process is sort of summarized like. You have a step where these nuclei are isolated to keep the chromatin intact. They are, sorry, they are exposed to TN5 transposase. As you can see, this enzyme sort of works like a homodimer. This green colored two subunits are able to assemble there. And then you isolate the transposed fragments and you amplify it. And then you sequence and identify any accessible regions which are present there, which will call for your ATAC-seq peak. So yeah, that's pretty much what we see here. Then here you see, yeah, so here you see transposition into native chromatin, and after transposition has been carried out, what you see on both ends of the region which is of your interest, or the region which is accessible and the region which you want to identify, at the ends of it or at the boundaries of it, the transposase was able to bind and it was able to induce nicks, which means single-stranded cuts. And these are TN5 induced or TN5 transposase induced. And after transposition, you can see the presence of these adapters, like we just mentioned, which are present there. And yeah, so and the I5 unique overhang is present on the left, while the I7 unique overhang at the right, shown in blue, generate the sort of post-transposition DNA fragment that we can now use further. Now, after there's an initial extension at 72 degrees centigrade, post-extension, there is amplification and addition of barcodes and adapter components, and then finally the purification of the entire ATAC-seq library. And the sites of chromatin accessibility get defined by TN5 insertion. So After the insertion of these PCR handles, the nicks which were left behind by transposase get filled in during the extension step that I just mentioned in the first step of the barcoding PCR. Then these fragments can be barcoded and then sent for sequencing. So this just shows you like a representative toy example of what hypothetical ATAC-seq data might look like. So you have two different cell type X and Y, both of which have ataxic peaks at the gene promoter. And however, cell type X has a cell type specific enhancer upstream of the TSS, which drives very high expression of gene A. In the ataxic data, this sort of appears as some cell type specific ataxic peak, which as you can see between X and Y can be quite different. In this toy example, the differential accessibility is driven by transcription factor B, which binds to the enhancer and increases gene expression. Then ataxic data can also reveal the binding motif which is associated with these peaks. Here that is represented as GATAA. So you can identify not only the region, but also the sequence of the motif which is allowing the recruitment to actually proceed. Yeah. Uh, Simplified sort of steps, if we look at them. Um, this is the TN5 transposase in just a little more in a more simplified sort of fashion. So like you can see here, there are two adapters present, and this is sort of an hyperactive form of the same enzyme. Yeah. And it has two different adapters on top of it, adapter 1 and 2, which are I5 and I7 overhangs. Like you can see, uh, then you have these crude steps, which includes cell preparation, which is obviously the first step, where cells are harvested, counted, and lysed so that you get your pure nuclei. And it's important to maintain single cell suspension so that you can get more accurate results. Then these cells get lysed in single cell suspensions. They're lysed by lysis buffer, which breaks open the cell, releases the nuclei, keeping the chromatin intact. Then these cells can be sort of centrifuged and washed to remove excessive force. The obtained chromatin is then fragmented. Yeah. The obtained chromatin is then fragmented and tagged with the sequencing adapters using the TN5 transposase to create the library. So the first step is your cell preparation, where you are lysing nuclei, keeping the chromatin intact, which you then centrifuge and wash. Then you fragment it and tag it using some sequencing adapters, like we just mentioned, which includes the transposase enzyme that helps in ataxic library generation. And then the library is purified, excess transposase and other contaminants are removed. This is the binding of TN5 transposases to open chromatin, fragmentation and tagging, or otherwise also known as tagmentation by TN5 transposase. So tagmented DNA fragments are those which have undergone transposition and have ligated adapters at both ends. Uh, just a second here. Then the next step you can see is amplification. So the purified DNA fragments, they undergo PCR amplification. What does that do? Obviously, that increases the amount of the quantity of DNA for which can be used for sequencing. Secondly, the final library can be checked using gel electrophoresis or fluorescence-based detection methods. Um, the final sequencing step. So what are the key steps? We use the nuclei, we isolated chromatin from it without disturbing the architecture. Next, we prepared ataxic libraries by following the fragmentation procedure. Then we did amplification using PCR. And finally, we sequenced it. Most commonly, the platform which is used for AttackSeek is Illumina, and paired-end sequencing usually is preferred because obviously it sequences both ends and provides more information about the fragment size and location of accessible regions. Single-end sequencing might also be used, but usually that gives less structural information and is less accurate. Um, yeah. Uh, what are the four main steps of data analysis? Pre-analysis, core analysis, advanced analysis, and multi-omics integration. Pre-analysis involves QC, sequence alignment. Again, this is pretty similar to the RNA-seq data analysis and the whole genome sequencing data analysis, like we've already discussed. You do an initial quality control to pre-assess or pre-analyze your data. Trimomatic is now used to remove low quality reads and low quality adapter sequences, after which trimmed reads are aligned to the reference genome. And after alignment, QC helps in filtering out those low quality reads, removing any duplicates, and checking the fragment size. Then comes your actual analysis. What does that entail? Of course, that entails calling of the peak, which will help identify any open chromatin regions. So most peak callers for um, attack-seq, they are adopted from ChIP-seq or DNA-seq method. There's one which is specifically only for attack-seq, which has this HMM attack name, hidden Markov model for attack-seq, this nomenclature. Um, yeah. Then just yeah. Um, Finally, then attack-seq, advanced analysis of attack-seq, it identifies the four key patterns that are of interest in the data output. First is the peak regions, which have come up with a lot of depth as regions under control. Second are motifs, which are consensus regions, which are able to bind, which are able to recruit proteins at that site. Then you have yeah, transcription factor footprint, and finally the nucleosomal constitution. It includes any annotating peaks, detecting transcription factor motifs, identifying transcription factor footprints, and studying the position of the nucleosome. Finally, ATAC-seq data can also be combined with other omic data sets to fully understand gene regulation. When we talk about multi-omics integration, it helps us understand how gene regulation proceeds in a much more detailed, much more nuanced manner. Um, ATAC-seq data with ChIP-seq is often used to confirm binding sites using RNA-seq to yeah, correlate chromatin accessibility with gene expression and reconstructing any regulatory networks. ATAC-seq requires much less number of cells, making it a great strategy for rare or less abundant samples. Then you have a simpler library preparation method, making it much easier and much faster to perform. No prior knowledge of epigenetic mechanisms is required. It has very high sensitivity, very accurately detects chromatin and regulatory elements at both bulk and single cell levels, provides really fast results, which make it suitable for large scale applications like clinical research and drug development. It can be used to understand diseases that are linked to epigenetic changes, that includes cancer, autoimmunity, or neurodegenerative diseases, etc. Where does it actually find itself limiting? So ATAC-seq can only capture the accessibility of chromatin and does not give any information about DNA modifications like DNA methylation, histone modifications, and chromosomal intergenic or chromosomal interactions. The TN5 transposase that is used in ATAC-seq, that is a modified engineered version, which can often lead to some amount of bias in the adapter ligation step. And sometimes it can insert the same adapter also to both ends of a DNA fragment. PCR amplification step can create challenges when dealing with large DNA fragments. And it faces a lot of computational challenge as only very few bioinformatic tools or analysis tools are available for ATAC-seq data. Usually ChIP-seq and MNase-seq data is more studied. Okay, applications first, of course, is mapping open chromatin regions to understand which regions are accessible for transcription. These help identify regulatory elements like enhancers and promoters that play very important roles. It has also been used to study gene regulation and transcription factor binding in different biological processes like complex diseases, immune responses, and cancer. It is also used in nucleosomal mapping, which helps to understand how this structure influences gene activity. Also, it can be used to study cancer epigenetics and see chromatin accessibility changes in cancer. It is also used to map disease-specific chromatin changes, and it also has application in computer genomics, epigenomics, and evolutionary studies. Just give me a minute. Okay, next we moved on to MNA-seq, which is micrococcal nuclease-seq, where 10,000 to 100,000 fresh or formaldehyde cross-linked cells are used for library construction. The digestion of chromatin by MNAs typically results in sort of a nucleosomal ladder. Yeah. Sort of a nucleosomal ladder, which consists of mono, di, and trinucleosomes and so on, depending on the concentration of MNAs. Usually that gives around 70 to 80% mononucleosomes and then higher order nucleosome ladders as well. The positioning and the occupancy of nucleosomes in the genome play very key roles in chromatin accessibility. It's an endo-exonuclease that leaves DNA regions, certain regions of the DNA, without nucleosomal protection, leaving it undigested. And it can be purified, adapter ligated, PCR amplified, and then sequenced. Therefore, it is an orthogonal assay compared with DNA-seq and ATAC-seq as it measures these regions and is most widely used as a method to map nucleosome positions genome-wide. Recently, quantitative protocol involves subjecting aliquots of samples to different levels of MNase digestion, allowing much clearer distinction of nucleosomal position and the occupancy from higher order chromatin properties, which can also be summarized sort of in a theoretical network. So there is a comparison between ATAC-seq, ChIP-seq, and MNase-seq. Next, we will move on to our discussion for today. So, what we are looking forward to today are methods for, these are also like sort of advanced uh, techniques for understanding gene regulation and understanding the chromatin, which is surrounding a gene. But we're going to do it in a different manner. We are going to look at chromatin accessibility profiling in a different way. What does that include? That includes 3C method. So we will look at that in details. Before we go, before we delve into all of that, we'll first understand the genome architecture and the 3D landscape of the same. So the human genome approximately has around 2 meters of DNA. Which must be efficiently packaged or compacted within a nucleus, which is less than 10 microns. And that is equivalent to packing 40 kilometers of fine thread inside a tennis ball, which is an unimaginable feat. And this process is not a random entanglement. Instead, it is very highly sophisticated, very dynamic, very essential for cell survival and cell functionality. Each cell must continuously negotiate. Each cell must constantly negotiate an equilibrium between the demand for extreme packaging and the critical need to access its genetic information for fundamental processes. So the amount of energy that will go into unpacking and the amount of energy which will go into maintaining it in that packed state, both of these are quite immense. So the cell continuously needs to make a decision between these two at every point of time. Um, the solution to this packaging paradox lies in the 3D genome organization. So instead of imagining the genome as a straight line, as a linear code, the genome actually exists as a functional, very folded, very structured landscape, which is hierarchically organized and begins with the confinement of individual chromosomes into localized regions, which are sort of called as chromosome territories. So if you look here in the schematic, this is your DNA, which has four histones, and around it, the DNA molecule is wrapped, forming these nucleosomes, which form chromatin fibers, form chromatin loops, chromatin domains or tags, and then you have chromatin compartments, and then you have chromosomes inside the nucleus. So the genome exists in this folded, packaged, and extremely regulated landscape. And this is organized in a way which begins with the confinement with individual chromosomes into these small volumes, which are known as chromatin compartments or chromatin territories. Within these, the chromatin further gets segregated into a large scale, very active compartment and an inactive compartment. At finer resolution, these are built from smaller self regulatory self-interacting regulatory loops. So if you look at like atomic scale detail, you would be able to see these stats, which are topologically associated domains. Topology basically means it's topologically in this case means that um, conformationally or structurally, this interaction has been made feasible, which otherwise might not have been so. So topologically associated or associating domains or tags are shaped by very specific point-to-point chromatin loops. So these are very tight and very organized. Um, This is, however, much far from the static or merely sort of structural. Um, it represents a critical layer of gene regulation, and by folding in these three dimensions, um, the genome can bring distant regulatory elements like enhancers and silencers into direct physical contact with the target gene promoters in an act that is fundamental to controlling gene expression. Okay. Yeah. So then the next question arises, why are we so invested in this genome architecture? Like, why is the information from the 2D genome not good enough, and why are we bothered about this 3D nature or the conformation of the entire genome? So the functionally, the importance of the 3D genome is illustrated when you look at the sort of adverse effects of it not being structurally compromised. So usually you would find that if there are disruptions in in this complex, it often leads to very diverse range of human diseases, from developmental diseases to cancer, etc. Now, chromosomal rearrangements, for example, which are a hallmark of many cancers, they do much more than simply alter the linear sequence. They can catastrophically rewire this whole thing. From the history of molecular biology, the 3D genome remained largely invisible, only accessible through microscopic lenses, and that has collectively been the driving force behind a revolution of 3D genomics. So, first described in 2002, the foundational 3C method, it provided a powerful sort of new logic which converted the transient physical proximity of genomic loci into stable, quantifiable gene ligation products. The conceptual leap which bridged the gap between physical structure and the genetic sequence, allowing researchers for the first time to see the folded genome. So that is the entire sort of selling point of these techniques, is that you don't just understand chromatin accessibility, you understand what the DNA actually looks like in 3D, what sort of interactions stabilize it, what destabilizes it, etc. So if you look here, the key steps can be shown in this following manner, where you have cross-linking occurring, followed by digestion or cuts being made at these loci, and yeah, and then ligation. Anyway, next you have on the right another representative example whereby you have these candidate, and with it you have these interacting fragments. Just sorry, there is some disturbance in my network. Okay, so I was saying that candidate interacting fragments are these within this region, and this is your constant fragment. This is obviously the genomic DNA, and after the 3C assay, yeah, after the 3C assay, what will you get? You'll have this Tacman Pro, which has a fluorophore at one end, some other product at the other end, and by qPCR elongation step, you will detach this as well, and you'll get a fluorescence readout every time that this amplification is taken place. So for 3C assay, qPCR elongation is good enough for final validation. Now we will understand how this happens in more detail. So, what are the common steps of 3D genome characterization, the chromatin accessibility profile? Protocol simply is intended to answer one question. Do two genomic regions that are distant in the linear sequence physically interact within the 3D nuclear space? So this is where the protocol is actually superior compared to what previous techniques were able to do. Like, for example, firstly, that's the concept of attack seek the assumption that your entire sequence is linear. Like on the right, what it looks like. This is how we usually assume the genome to look. And from here, whatever data you were getting was representing that, oh, an X region is accessible, B region is not, or if under some variant condition, some treatment or some experimental difference, if we see a shift in that chromatin profile map, then that can be read or that can be considered as a readout. So um, in this case, in case of chromatin or chromosome conformation capture, here what you're doing is you are converting a biophysical property, which biophysical property? Spatial proximity. So these are two regions that are being that are under scrutiny to understand how proximal they are to each other and convert this biophysical property into a genetic one. So a novel artificial DNA sequence at a ligation junction that can be detected and quantified. So this creates a permanent linear record of a transient 3D interaction based on which all the subsequent high throughput methods are built. So what do we mean by that? What does it assume? It assumes that, or not assumes, or it considers that if there is spatial proximity. Yeah, if there is spatial proximity, can we convert that into a genetic one? So what you do is you have a novel artificial sequence that is detected, that is quantified, and you create a record of the transient DNA interaction. How do you do so? We'll learn about that. Briefly, if you look at the schematic on the right, you'll see that the first is, of course, cross-linking between the DNA and the protein. Then you have the digestion. What part is digestion going to play here? It is going to allow ligation to proceed in the immediate next step. So right after digestion, what you see are the two ends, two open restriction digested ends undergoing a ligation process, and finally detection using PCR. So following cross-linking, sorry, let me just read this first. So if you look at the 3C protocol, it's sort of a multi-step biochemical process also that also requires a lot of careful execution. First step is in vivo cross-linking. So you have intact living cells. You use a cross-linking agent very similar to what you do in case of ChIP-qPCR or ChIP-seq. There also we sort of freeze the DNA protein interactions in place. And so formaldehyde is used, which penetrates the cell and the nuclear membranes. And what does it do after penetration? It creates covalent DNA protein and protein-protein cross-links. Is the idea behind it? The idea is that you want it to be very similar to having a high resolution snapshot. What do we mean by snapshot? We mean basically that all of the DNA protein interactions inside the cell are just frozen in space and time, and you have clicked that in that moment, at that point of time, this is what the interaction looked like. Now, somebody might ask, why? Why are we doing that? Isn't that going to be biased? Well, it would be wrong to say a very hard no. But what you can think of it as is that if you consider the entire experiment, then throughout the protocol, a lot of conditional variation is expected to come about. And to ensure that the change in your chromatin conformation or chromatin accessibility is not the consequence of those experimental variations, and instead is something that was already present in vivo inside the cell at that point of time, to make sure that it's not an error, but instead the norm or the physiologically, biologically valid um, conformation of the chromosomes. Therefore, it's essential to freeze such an interaction or freeze such a state. So formaldehyde is able to do that job. And careful standardization of formaldehyde cross-linking is extremely important. Okay, if any of these things, if you want to read about them in a little more detail, you can refer to any sort of review article, etc. And apart from that, you can also look at CD Genomics, this website. They have it's sort of a portal which can also be referred to, and there you'll find a lot of this study material already available. So yeah. So following cross-linking, the cells are lysed, nucleus is isolated. So this is sort of a part of your cell preparation step. This cross-linked chromatin has then been digested with a restriction enzyme such as Hindi3 or EcoR1. So after digestion, what does that do? It creates these compatible ends. So think of it like this end, for example. If you look here, it will be difficult to ligate these ends because they might be completely different. But instead, if I have two ends which have been digested with the same enzyme and therefore the ends are compatible, they have nice overhangs which are able to readily ligate to each other, then that would be a more favorable sort of condition that includes enzymes like Hindi3, CoR1, etc. And it obviously cuts DNA at very specific predictable recognition sites, which creates a very complex library of chromatin fragments. Crucially, fragments, just a minute. So crucially, fragments that were spatially proximal in the nucleus remain physically tethered together by the network of cross-linked protein complexes. So till now, what did you do? You isolated the nucleus. Here. So you isolated the nucleus, then you prepared your cells, you did formaldehyde cross-linking, isolated the nucleus, did chromatin fragmentation, and following that, you did chromatin interaction linking. This is the conceptual and technical core of the 3C method. What is done here? You have your cross-linked digested chromatin fragments, which you then subject to ligation with DNA ligase under extremely diluted conditions. What does that do? It gives you a very high volume of the reaction mixture that ensures that chromatin complex concentration is very low. So basically, you are giving very limited, very deprived and unfavorable conditions for the cell to be able to carry out that ligation protocol. But despite those limiting conditions, despite those limitations, the probability that two fragments from very different complexes randomly colliding and randomly getting ligated, which means an intermolecular ligation occurring, that probability decreases by a lot. Instead, what will happen, because you have diluted conditions and because you have reduced accessibility, Therefore, what happens in this case is that fragments which are very close to each other, they will actually preferentially get ligated with each other, and this captures true three-dimensional interactions, creating very novel chimeric DNA molecules where the junction represents a point of spatial contact in the original nucleus. So this is what you see here, a representation of sort of these chromatin bubbles or chromatin territories inside the nucleus. And if you cross-link chromatin using formaldehyde like chemical reagents, then you do a restriction enzyme digestion, then you'll be able to generate these little fragments which are in place because they are in close contact with each other. Just yeah. Yeah, so these are in contact with each other. And if you see, restriction site digestion has also taken place. And after restriction enzyme digestion has occurred, if you ligate it with an antibody, sorry, if you allow ligation to proceed and then you reverse cross-link, which means you open up this DNA protein interaction and instead you have these complete loops, then you can do a PCR sort of a reaction. So after ligation, the cross-links need to undergo reversal. Typically, that's done by heat, treating with proteinase K, which sort of digests the protein which was holding these two fragments together. And then you degrade the protein, you release the DNA. This resulting DNA contains a mixture of some religated original fragments, which means that these were original fragments that have just gotten sort of ligated to each other, and you'll have some chimeric ligation products of interest. Now, the original TC protocol used to use qPCR with a pair of primers that were able to specifically amplify the junction between two predetermined genomic loci. The amount of PCR product generated will then be measured, providing a quantitative readout, which is directly proportional to the frequency with which those two loci interacted in the original cell population. So how much PCR product is generated can then be measured, which will give you this quantitative readout. So yeah, so that's what was done in the original technique. So you have 3C, which is a 1 versus 1 contact. You have 4C, which is 1 versus all possible contacts. 5C, which is many versus many. Don't worry, we'll learn about this in the next few slides. Kaya PET is for all versus all in contact proteins. High C is all versus all again. So all of these techniques, they give you different sort of information. We'll learn more about these methods in tomorrow's class, Kaya PET and High C. Today, we are keeping it to these chromatin conformation associated methods, which are chromosome conformation capture methods. So, yeah, so that involves some sort of sequencing here as well. So this diagram shows you the common initial step for all of the methods. You have formaldehyde cross-linking, like we already mentioned. You have restriction enzyme digestion. You have chromatin interaction linking to create a 3C library, which then branches to show you the unique features or the unique steps for each technique. In 3C, you use specific PCR primers to detect an interaction between two known fragments. 4C, that involves something different. Let me just go to this technique. It will make it easier. So this is your cross-linked chromatin, which is digested using some enzyme. The ligation is allowed to proceed to link these two regions. And these are sort of chimeric DNA molecules. Then you reverse the cross-link and you'll have this newly joined sequence. Now, the original strategy, like we just mentioned for 3C, that involved semi-quantitative real-time PCR-like methods or just some semi-quantitative PCR method. Then you have 4C, in which case things are a little bit more different. In 4C, you have digestion with a restriction enzyme, followed by ligation to generate a completely circular product, which is then followed by inverse PCR from some known fragment, which is your bait fragment, to identify all the interaction partners. So if you look here, this 4C method is one versus all, which means all of the interaction partners can be identified in 4C. Then you have high C, which incorporates biotin labeled nucleotides during the fill-in step. Let me first explain 5C in this case. In 5C, you use a multiplexed ligation mediated amplification strategy with universal primers. So if you look here, this is your 5' T7 and 3' T3 associated primers, which are able to bind ligation and PCR proceeds, and then you proceed with microarray or sequencing protocols. Then, if you look at high C, like I just mentioned, it incorporates these biotin labeled nucleotides during the filling in step, allowing for the enrichment of ligation junctions, and subsequent genome-wide sequencing is carried out. So this is high C, as you can observe that after shearing, you have antibody binding, enriching for those fragments which have bound, and then you add the adapters and you perform PCR or sequencing, and you'll get your results. Then coming to chip loop, just a minute. Okay. Yeah, sorry for the disturbance. We'll go through a video also, so that'll clear up more of these details, I think. But if you look here, we were talking about chip loop and QIAPET. I'll not go into details about that today because we'll learn it better tomorrow. But chip loop, it involves, again, the initial steps are similar. You have this you precipitate chromatin or you isolate chromatin, you digest it after cross-linking, then you ligate it, and you have these open sort of loop ends, and you ligate them onto beads, after which reverse cross-linking is done, and then semi-quantitative PCR is done. And for QIAPET, you add adapters and ligate it onto beads. And the difference between these two is that here you have a closed loop, here you have open loops, and then you reverse cross-link it, digest it, and use adapters and PCR, and you get all possible interactions. So this gives you way more information. So high C and QIAPET give you all versus all interactions. These give you, like, they give you a complete picture of the entire genome, versus when you do these other strategies, these have some step which gives you one versus one or many versus many form of data or form of information. So this QIAPET, like I mentioned, it features an immunoprecipitation step to map long-range DNA interactions similar to Hi-C. In this method, DNA protein complexes are cross-linked and fragmented. Specific antibodies are used. Also, before we proceed, QIAPET stands for chromatin interaction analysis by paired end tag sequencing. And yeah, this will be two sets of linkers with unique barcodes are ligated to the ends of DNA fragments, which are in separate aliquots, which can then self-ligate based on proximity. These DNA aliquots are precipitated, restriction digested, and sequenced. Deep sequencing provides some base pair resolution of these ligated fragments. Hi-C and QIAPET currently provide us the best balance of resolution and reasonable coverage to map long-range interactions. So what is the advantage? First, it gives us the ability to detect a large number of both long-range and short-range chromatin interactions globally. Um, Wait, let me just give me a moment. Yeah. So let me just show the video. I hope it's audible. Let me check. Just excuse me a minute.
That are used to identify chromatin-chromatin interactions.
And so that concludes our discussion of the DAM-ID assay. And so we'll now move on to different techniques used to measure interactions between genomic loci. And so I mentioned that there's a rather large number of such techniques. So some of them include 3C, 4C, 5C, Hi-C, and QIAPET. We'll mainly talk about the Hi-C assay. But generally speaking, all of these assays are predicated on the idea that if you cross-link DNA such that genomic loci that are physically proximal to each other get cross-linked together, then the cross-linking results in them basically being stuck together, and you essentially form a read out of sequences from both genomic loci that are stuck together, and the mapping of those reads to the genome tell you essentially which genomic loci were in close physical proximity.
So the basic question is, why do different genomic loci that are potentially distal to each other on the linear chromosome end up being close in physical space? And so the first reason is that there could be like protein-protein interactions that come from, for example, enhancer-promoter interactions. So for example, transcription factors that are bound at enhancers at some point typically need to physically interact with factors that are present at the promoter of a target gene. And so when that happens, obviously something like chromatin looping has to happen. And second way you can get close proximity is that you can have what's called bystander interactions, where it's not that two genomic segments are actually in physical contact for functional reasons. They could just be in close proximity because they're nearby other sites that are bound. Third reason is that because chromatin is, you could essentially think about it as like a really long polymer chain. That means that genomic regions that are close in linear sequence will also tend to be physically close in space in the nucleus. And so you'll get a lot of like non-specific interactions between physically close genomic loci. And finally, because of interactions with the fixed nuclear landmarks, like the lamina, you can get close proximity of two genomic regions just because they happen to also be, for example, contacting the same nuclear landmark. Again, not necessarily because they're actually physically interacting per se. So most of the technologies that people use based on sequencing to detect interactions between genomic loci are based on what are called 3C technologies, or chromatin conformation capture. And so 3C based technologies include what you might call like the classic 3C approach, 4C, 5C, chia PET, and high C. And so all of the 3C based technologies share a common step for generating a chimeric DNA fragment. And so the general idea here is that if you take your native chromatin and you cross-link with, for example, formaldehyde, so recall that cross-linking can cross-link proteins to other proteins or proteins to DNA. And so after cross-linking, what you typically do in these 3C technologies is that you then digest using some restriction enzyme that cuts reasonably frequently. So something like HIND3 is a classic restriction enzyme used for 3C technologies, because it's what you call a 6 cutter. So its recognition sequence is 6 base pairs. And so that means that it cuts every about 4 kilobases in the genome. And so what you end up with after you use your restriction enzyme is that you'll, generally speaking, end up with a lot of fragments, including basically distal genomic loci that are cross-linked together because of, for example, protein-protein interactions that are present where one protein is bound on each fragment. And so what happens is that after you digest with the restriction enzymes, you then perform a ligation step where basically the free ends can ligate together from the 2 distinct loci. And then after you reverse the cross-linking and purify, then what you end up with in principle is a lot of what's called chimeric DNA fragments, where part of the fragment comes from one genomic loci and part of the fragment comes from the other. And so what's important to note here is that here I've indicated that at the terminal ends of the chimeric DNA fragment, as well as in the middle, you basically have HIND3 restriction sites because HIND3 was used in this case for fragmenting the genome. And so these HIND3 sites are going to be important in the later steps of describing the 3C technologies.
So the classic 3C approach is what's known as a one-to-one region approach. And so the idea here is that after the cross-linking, the reverse cross-linking step that I talked about in the last slide, basically all you need to do is anneal sets of primers where your primer set consists of primers targeting regions from locus 1 near the known HIND3 restriction enzyme sites, and also from locus 2 near the restriction enzyme sites of HIND3. And so because your primers are specific to regions near the HIND3 cut sites in locus 1 and locus 2, then you're basically only going to amplify fragments corresponding to regions from locus 1 and locus 2 that are in close proximity. And so because your primer sets are specific to each locus, that means that realistically your primer set really only covers like a few hundred kb for each of the 2 loci. And so again, 3C, the classic version anyways, was a one-to-one or one region against one region type approach. It's worth pointing out that more recently people have kind of revisited the classic 3C approach and modified the approach in order to produce a protocol called 3C-seq. And so the idea of the 3C-seq approach is that similar to the...
This is something that should be noted, that after 3C, instead of opting for a very restricted conclusion from such an elaborate experiment, currently people are sort of switching to this strategy, which gives you a lot more output with the same initial protocol or initial processing step. So moving on.
4C based approach that I'll talk about on the next slide use a more frequently cutting enzyme, something like DPN2, that'll produce shorter fragments. And then what you do is you directly ligate sequencing primers on the terminal ends of the fragment, and then you just kind of sequence all of the fragments. And so in theory, your chimeric reads can come from anywhere in the genome, not just the ones that you've designed specific primers to. The next approach we'll talk about is called 4C, or what's known as circular 3C. And so 4C is an approach that's known as one against all, because you're really assaying for interactions between one locus against the rest of the genome. And so in the 4C protocol, basically what you do is after you reverse the crosslinks from the 2 slides ago, basically the idea is that, again, you use kind of like a more frequent cutter, what's known as like a 4 cutter, such as DPN2. And so DPN2 basically has a recognition site of 4 base pairs, which means that it cuts every 256 base pairs or so. And so the idea is that after you cut with this secondary cutter, like DPN2, you end up with a shorter fragment that's still chimeric. And you can basically circularize this fragment. And then what you do is you, similar to the 3C, the classic 3C approach, you then anneal primers that are specific to your target locus and do the difference with 3C is that your primers are actually still only targeting one locus. That's your locus of interest. But and this time also your primers are targeting not just sequences near the HIND3 cut sites, but also the DPN2 cut sites. Because basically your chimeric fragments should have one HIND3 site and one DPN2 site. And so basically by targeting both ends of the fragment corresponding your target locus of interest, you're going to again get these chimeric fragments, which you can then sequence, and then through mapping, you can then figure out which other regions in the genome are interacting with some locus near your, or are interacting with some fragment in your locus of interest.
Okay, so that is pretty much what I wanted to cover for today, but cover in this particular video. But you can watch more videos by this channel to get more clarity over this technique. But basically, what I wanted you to learn were two things. First, understand what exactly is the goal of using 3C. And first of all, get introduced to this method. It is not something people are usually very familiar with. So understand 3C method, understand its utility, its caveats. Um, yeah, apart from that, I think, yeah, I will not elaborate too much on this today. I'll just introduce you to the basics. Um, just a minute. Okay, so yeah, so Kayapet is a different strategy altogether, but it maps long-range DNA interactions similar to Hi-C. So DNA protein complexes are again cross-linked and fragmented. Specific antibodies are used so as to immunoprecipitate or pull down your protein of interest. And then you use linkers, which have unique barcodes that you ligate to the end of your fragment in different aliquots, which can then self-ligate if they're close to each other. Then the DNA aliquots are precipitated, digested with restriction enzymes, and sequenced. Deep sequencing provides us base pair resolution of these ligated fragments, and Hi-C and Kayapet currently are the most informative in terms of resolution and coverage to map very long range interactions. Now, what are the advantages? It is very useful when you want to detect large number of long range and short range chromatin interactions globally, if that is your goal. Now, when would you consider that your goal? For example, if your gene of interest or if your treatment or if your condition is something that has a role in controlling chromatin interactions, then that is one thing. If it studies the interactions made by specific proteins or complexes, then that is another advantage. Public QIAPET data sets are available through the ENCODE project, which removes background generated during traditional chip assays. When you perform immunoprecipitation, that sort of narrows down the data complexity also to a large degree. Cons: first, you need an immense amount of starting material, at least 100 million cells. That is 10 to the power 8 cells, which is quite a big number. Secondly, if you have non-specific antibodies, any unwanted protein complexes potentially can get pulled down and can contaminate your data pool. Linkers sometimes are capable of self-ligation. Some ambiguity might be generated. Very little sensitivity might be there. Sometimes might detect as little as 10% of the interaction. So the information which you get out of it sometimes can be much less than the actual information which is there. So um, that was about QIAPET. Then we talked about high C being an all versus all strategy, which gives you a very global, comprehensive perspective. The 4C and 5C techniques, they represent two very distinct and very evolutionarily very powerful branches from the original 3C methods. They are not just predecessors to high C, but they till date are extremely relevant, much more cost effective because as you can understand, the scope that or the umbrella that high C as a method actually encompasses is immense. And so cost effectiveness is something that needs to be kept in mind for high resolution hypothesis driven research questions. And they do certainly fill a crucial niche by providing deep targeted data at a fraction of the cost, which is required to achieve much similar resolution with genome-wide methods. When we talk about 4C, then the viewpoint analysis, the 4C technique was developed to overcome the primary limitations of 3C by enabling an unbiased genome-wide search for all sequences interacting with your locus of interest, the bait. The 4C workflow begins with generation of a standard 3C library. The key innovation occurs that after the first interaction linking step, DNA is then purified and then subject to a second digestion with a different, more frequently cutting restriction enzyme, which is DPN2. Now that gives you much smaller fragments. They can be ligated again under very dilute conditions. That part remains similar, which is designed to promote cell circularization. And as a result, you get very tiny circular DNA molecules where each circle ideally contains the known bait fragment ligated to a previously unknown interacting prey fragment. To identify these, inverse PCR is then performed. So what is done? You are getting very tiny circular DNA molecules. Then inverse PCR is performed where you have a bait sequence to which your primer binds, and then outward amplification is done across the ligation junction and into the unknown sequence. And then that can be quantified by hybridization to microarray or by sequencing. So again, if this was confusing, just quickly I'll show you this image. So if you see here, um, I don't think the method is very clearly shown here. Yeah, here inverse PCR has been mentioned, but kind of think of it like this, that outward PCR is carried out. Like for example, here, outward PCR, there's a circular molecule. So in this way, the sequencing PCR will be completed, and this region of your interest will then be sequenced and can then be decoded. So the resulting library of amplified fragments can then be identified. Prey fragments can then be identified and quantified by hybridization to a microarray, in which is why it was considered to be on chip, or what is currently 4C-seq by performing high throughput sequencing. So overall, this is what it looks like. The 3C template, you have your chromatin loop cross-linked by some protein of interest. You cut it, fragment it using the first restriction enzyme. You ligate it in vitro, remove cross-links, purify, and then sequence it or study it. Uh, perform qPCR, for example. For 4C, you do the first step, then you trim it using the second restriction enzyme to get a chimeric DNA sequence. Then you ligate it among itself so that it becomes circular in nature. You create a library of this form where you can perform inverse PCR using primers, which are able to bind to your base sequence, which are able to bind to a known sequence, your base sequence, and then proceed forward with the amplification step. You generate a library of such fragments subjected to NGS, and you quantify what captures have been obtained to get an idea that these are regions which are close to each other. And the 3D conformation of the chromatin is such that it has brought regions from these two loci close to each other. So the principal application of 4C-seq is to generate a very high resolution one-dimensional interaction profile that maps the entire regulatory interactome of a single viewpoint. This makes it an invaluable tool for discovering novel long-range enhancers that regulate a gene of interest, for identifying some target genes. If you have long range enhancers that regulate your gene of interest, that can be one possibility. If you have target genes of non coding disease associated SNPs, or want to characterize how the nuclear environment of a specific locus changes during cellular differentiation or in response to stimuli, you can do that. For example, 4C-seq has been instrumental in mapping the complex interaction landscapes of oncogenic enhancers, revealing how they hijack regulatory control over distant genes in cancer cells. So that is what will sum up the discussion I wanted to do for today. So just to quickly summarize our discussion for the day, let me just switch on my camera. So issues. Okay. Yeah. So basically, what we understood today, first of all, we understood the fact that the chromatin architecture makes a lot of difference into affecting how any region can be transcriptionally controlled, whether or not it will be expressed, whether or not it is controlled by some random gene which you might not even think of, but is from a distance as a long range enhancer working on this execution. Uh, Secondly, how does it supersede what we already knew? So, for example, if you consider ChIP-seq data, then what it tells you is that there is a direct binding of some protein onto your region of interest, or close to your region of interest, but you cannot always be sure that these two regions are um, physically close to each other in the chromatin, and they're able to regulate this sort of interaction. If it is a long range interaction, if it is an upstream enhancer that it actually binds to, then that can be a more interesting area of study as well. Um, where is it relevant? It is relevant if your gene of interest is a DNA binding protein, and if you want to validate its interaction with some other protein, which is also DNA binding, but not a transcription factor, but is instead a regulator. Either it is an activator, or it is a chromatin remodeler, or it is some sort of a scaffold protein which is able to do this. So all of this, a scaffold protein directly cannot be studied. Of course, you need to have a DNA binding protein. Otherwise, it'll completely go unrecognized. So opportunities are endless. There's so much to learn. I think it was 2006 or 7 when 3C was first discovered. And of course, that has gone through multiple rounds of iterations, multiple rounds of advancements, of modifications that make it A, more reliable, B, more accurate, and C, more accessible and affordable in today's time. And C, also, these techniques, these have undergone multiple levels of modifications to make sure that, considering the investment that is made into this technique, and by investment, I don't just mean monetary investment, but in terms of the number of cells that you have used. Growing cells also costs a lot. It is a monetary investment. If you're using antibodies, it not only uses um, uh, like a financial burden is not only associated with it, but it also involves animals which have been sacrificed to generate that antibody, which have been used for for science. So considering those ethical parameters also, it's important to make sure that whatever resource utilization is being done, it can be redirected to give you the most informative and the most comprehensive results. And also what it also contributes to is to make sure that your results are not one dimensional in nature. You can truly understand the chromatin, truly understand how that works. So all of that sort of needs to be taken into consideration and needs to be appreciated and needs to be recognized. And yeah, so these are some really cool techniques that I wanted to discuss with everybody. And we've discussed 3C today, 4C today, a little bit about 5C. I've not gone into details about it. It's sort of like an upgrade on this technique itself. We'll discuss QIAPET and mainly high C QIAPET, and maybe a little bit about QIAloop also in tomorrow's class. So that should like sum up these chromatin conformation associated methods. And maybe I'll also discuss how you can actually get access to this data, where you can look at it and what can you do with that data. Maybe we'll go into that as well. So yeah, that's the plan for today and tomorrow. Thank you so much for joining. I hope this course has been useful. It's been a really long course. It has not only been a learning curve for you, but for me as well. So thank you for joining, and I've had a great time. I hope people who have been a part of this course have had too. I know it's been a little discontinuous, but I'm also a student and I am also studying and working and also trying to take classes. So it does fluctuate a little bit from time to time, and I apologize for that. But thank you so much. I hope it's useful. I hope you learn a lot more. I hope you make use of all of the resources which scientists around the world have built for you and me and everybody who is so in love with science, so in love with understanding how nature works, how everything inside a cell works, how biology works, and we do our best and we do our bit to make it a little bit better. So thank you so much. I'll close today's class. So have a good night. Bye bye.